So we did glycolysis. We took our glucose molecule, we split it in half. That gave us 2 pyruvate molecules. We oxidated pyruvate, and now we are ready to talk about the citric acid cycle. And just as a heads up, when you're going and studying this material and using other sources, you're going to see the TCA cycle, which means the citric acid cycle. You're also going to see it called the Krebs cycle, which is named after the scientist that described this process. Okay. But for the sake of our class, I will always call it the citric acid cycle. This material is tricky enough. I don't need to try to trick you, but I did want to give you guys a heads up so that you're not confused about what the TCA is or what the Krebs cycle is. It is the citric acid cycle. This is important. The students always get tripped up on this, so I'm warning you now. The citric acid cycle happens twice for every glucose molecule. What we're going to do is we're going to take that acetyl group. Remember that 2 carbons that are attached to each other. And we are going to turn that into some carbon dioxide. We are going to we're going to create some more NADH and this other kind of electron carrier called FADH2. So here it is, beep. Okay. In your lecture notes, you can use this link for a little animation from your textbook about the citric acid cycle or the Krebs cycle. Okay, let's do a quick overview. Okay, our acetyl-CoA is going to come in, and that is going to bind our 4-carbon oxaloacetate. There's a boy that you should know. So we're going to have 4 plus our 2 carbons from our acetyl-CoA, and that's going to give us a 6-carbon molecule called citrate, which is also called citric acid, conveniently enough, because this is the first stable compound in the citric acid cycle. Okay, the first thing that we're going to do is take some of our NAD+, convert it to NADH. We're going to pull off 1 carbon. That means that we're going to have a 5-carbon molecule left. We're going to do that again. NAD+ is going to be converted into NADH. As we pull off 1 carbon, now we have a 4-carbon molecule. We're going to produce an ATP molecule. Woohoo! And then we have all these 4-carbon molecules. As we rearrange all the other atoms that are attached to those carbons, we're going to be able to oxidize some more and produce some more of our energy or our electron carriers. The citric acid cycle takes place in the matrix of the mitochondria. So if here's my mitochondria, the citric acid cycle is taking place inside of the inner membrane, which is called the matrix. Okay, we are going to take this 4-carbon sugar called oxaloacetate. If you've ever heard or have used Barkeeper's Friend is oxaloacetate. We will see this chemical again in our next chapter. So he's a good one for you to also write down and remember. Okay. So I'll put a little star next to the things that we need to remember. Okay. Oxaloacetate is 4 carbons. Our acetyl-CoA, so here's our 2 carbons that were left over from our original glucose molecule. It's coming in, riding on that CoA. It's going to bind with our 4 carbons plus our 2 carbons is going to give us our 6-carbon citric acid. Okay. Now, this is also important. If we have enough ATP, this is the step where we are going to shut down, because we don't want to produce a whole bunch of ATP if we don't need it. ATP has about a 3-second shelf life. So if we're not doing anything that requires ATP, we don't want to be producing it. So when you're being a couch potato or you're sleeping, you have a lower energy needs, you're not going to be producing as much ATP as when you're running a marathon. And that any sugars that come in during the time when we are not requiring ATP, that glucose then is going to be produced into fatty acids, so that we can put that into the long-term storage. All right, now this is the process of where we are going to oxidize some things. We are going to take our citric acid. We're going to rearrange some atoms. Nothing cool is happening yet. Now, at this stage, we are going to take this 6-carbon sugar over here, and we are going to transfer some of the electrons from our 6-carbon sugar onto NAD+, and that creates our NADH. This is going to pull off. This is going to become our CO2. Now, what we can do from here is take this, pull those electrons from that bond, give them to NAD+, make some more NADH, and produce a carbon dioxide. So far, we've produced 2 carbon dioxides and 2 NADHs. So the next thing that we're going to do is we're going to take that coenzyme A. We're going to pull that off. As we pull that off, that's going to allow us to produce ATP. Okay. That's another substrate phosphorylation event. Okay. Now what we have left is a 4-carbon sugar. Eventually we need to rearrange those atoms so that we can get back to oxaloacetate so that we can continue the cycle. So what we're going to do as we move around atoms, rearrange some things, we are going to transfer those electrons. We're going to reduce FAD, it's another electron carrier, does the similar job as to NAD+ and NADH. So FAD is going to grab these electrons, become reduced to FADH2. We're going to do some more rearranging. Malate is another 4-carbon sugar that we're going to see again. Okay, we're going to take that malate. We're going to produce some more NAD+ or NADH from our NAD+, and that gets us back to oxaloacetate. Now we're ready to do the cycle again. Now, when I was an undergrad, I had to memorize the names of all of these chemicals, and the names of all of the enzymes that did that. I'm not going to do that to you. There are some things that I want you to know, so let's talk about what all of those things are now. Okay. Let's remember that the citric acid cycle is going to happen twice for every glucose molecule. Okay, so we started with our 6-carbon sugar back in glycolysis. I need one more. We broke it down. We pulled off a carbon during the oxidation of pyruvate. These are the 2 carbons that are going in. So this set goes around, this set goes around. So let's talk about our high energy molecules first. Okay. We have our reduced carrier NADH. We've got 3 NADHs because we're doing this twice. That gives us 6 electron carriers of NADH. We also get another electron carrier of FADH2. because we do this twice, that gives us 2 of those. We end up with 2 ATP molecules. Okay, these are our energies. Let's talk about the other parts of our equation that we see. We see a carbon dioxide as a waste product. Carbon dioxide as a waste product. We're going to do this twice. So that's 1, 2. Second round, 3, 4. Gives us 4 carbon dioxides. Here are the names of the chemicals that I need you to remember. We need to know oxaloacetate. We need to know its name, and we need to know that it's a 4-carbon sugar. You do not have to memorize the chemical structure unless you want to. We need to know acetyl-CoA. This is a coenzyme that's carrying 2 carbons. We need to know that citrate or citric acid is a 6-carbon sugar. We also want to know alpha ketoglutarate because, spoilers, this is where, when we break down proteins, this is where the step at which that enters the citric acid cycle. We're also going to want to remember malate as a 4-carbon sugar because we're going to see that again in the photosynthesis chapter. Also, since I told you I spoiled where the proteins come in, I'll spoil where the lipids come in. And the lipids, when we break down fatty acids, which are lipids, it's going to enter in at this acetyl-CoA state. Okay. With your guided notes, you're going to flip over to part 2 and answer question number 3. Here's a challenge question for you. We said that carbon dioxide is a waste product. Does carbon dioxide have any more stored chemical energy that cells can use? And I have a hint for you here of Diet Pepsi and Diet Coke. I want you guys to think about that. See if you can come up with your answer and be able to defend your answer. All right, let's summarize what we've done. During glycolysis, we took our 6-carbon glucose molecule, broke it in half, made our 2 3-carbon pyruvate molecules. We then, during the oxidation of pyruvate, we then produced 2 carbon dioxide, went into the citric acid cycle. You're going to do the citric acid cycle twice for each glucose molecule that we have. So now let's keep track of what we've done so far. We used our only glucose molecule way back during glycolysis, so we don't have to mark anything here. We still haven't used our oxygen yet, but remembering that we do the citric acid cycle twice, we have produced the 4 remaining carbon dioxide. So we've actually completed and produced all of the carbon dioxide that we're going to produce during cell respiration as a result of the oxidation of pyruvate and the citric acid cycle. We've not produced any water yet. And we have now produced 2 ATP molecules, 2 of these high energy electron carriers, 2 FADH2s, and 6 of our NADH. Okay, remember that these electron carriers are both in their reduced form, meaning that they are carrying around high energy electrons.